in feed could be used to improve feed intake of the lobster. Feed intake of pellet diet decrease when the lobster feed pellet diet with co-feed with the mixed fresh food. However, the total feed intake increase when compared to the only pellet diet. Feed intake of lobster when they feed a fresh fishery diet with the different composition. Feed intake of lobster feed mixed fresh fishery was lower than the feed or either only mussel or crab. And the lowest fish intake found in the lobster feed with the fish. Next, please. I think that's all my presentation. Thank you very much for your attention.
Thank you very much, Pak Nyoman. Very great presentation. So we have a time for discussion. I invite one participant who will have question probably or suggestion for the three presenters. No? Okay. Yeah. If there is no questions or suggestions, please give a reaction, applause, yeah, for the presenter. Thank you very much, Bu Dewi, Pak John, and Pak Giri for your presentation. And for next presenter, I will invite Ibu Haryanti.
Yes.
Bu Haryanti, ready?
Yes, I ready.
Okay. Bu Haryanti will present the effect of feed supplement on the recovery of operculum deformity in Baramundi. Lattes feed. Okay.
Yeah.
Bu Haryanti, time is yours.
Yeah, thank you, Mr. Chairman. Good afternoon, everybody. This afternoon, I will present the result of my research and title the effect of probiotic and vitamin C as feed supplement on the recovery of operculum deformity in Baramundi Ratus River seed. We have many scientists involved in this research. Next, please. Yeah. It is now that Baramundi is a fish that has high economic value and is very strategic for the global market. As you know, the people like to eat Baramundi fish, and the high demand for Baramundi fish is in the form of fillet, especially must be balanced with cultivation. However, the main problem in seed production and cultivation is disease infection, including viral infection, bacterial infection, and deformity. So the effort are needed to produce healthy seed of in Baramundi fish. Therefore, the production of Baramundi fry with the right quality and quantity is needed. Next, please. In fish fry, the deformity that often occur in skeletal deformities like scoliosis, kyphosis, lordosis, jaw malformation, and deformity in the gill operculum or shortness or shortened operculum. Several factors affect the deformity is environment, water source, temperature, microbial balance, feed, include natural and formulated feed. Disease also very affect the contributed to deformity. And the last one is genetic factor from broodstock contribute greatly in seed quality. That's why probiotic and vitamin C as a feed supplement is used to recover the operculum. Next slide, please. The objective of this research is obtaining information on the effectiveness of using feed supplement, in this case is probiotic and vitamin C, on the recovery of operculum deformity in Baramandi, Latus Calca River. Next slide, please. Yeah, the method of this research is we use polycarbonate tank 200 liter, and we use a piece with body weight, body length is around 90 centimeter, and the body weight of 4 centimeter. And the treatment applied to fish without deformity or normal were fed on commercial diet. Fish with deformity, we feed with the commercial diet, and sea treatment is fish with deformity and fed on commercial diet supplemented with probiotic of Saccharomyces cerevisiae and mixed with vitamin C. And all treatment repeated for replication, and we use water exit around 4 until 50%, and observe for this result is growth, survival rate, level of deformity recovery, genetic variation, use RAPD method. Next slide, please. Yeah, the result of this research is like in this slide, the group of length of Barramundi juvenile during rearing in different treatments was not significant difference, but on body weight growth profile saw the significant difference. And survival rate, next slide, please. Yeah, and survival rate was not different also among different treatment, between 90 until 97%. Next slide, please. From this calculation of the average percentage of Barramundi fish that recover from deformity in treatment B, saw that 11% of the operculum had changed to normal and close properly. Some still in recovery process at 21%, and still deform 68%. In treatment C, not yet, yeah. In treatment C, where the Barramundi fish feed with supplemented feed, probiotic and vitamin C on micro diet, the average value of the normal operculum was 20%, while some of seed were still in recovery process at 14% and still deformed at 66%. This result revealed that the supplemented probiotic and vitamin C should recovery deform operculum in Barramundi. Next slide, please. Yeah, this one is similar with former slide. The study showed that normal treatment for A had deformity 1%, fry deformity, and and treatment B was changed normal 11%, recovery process 21%, and still deformity 68%. While the fry deformity with feed supplement C treatment was changed normally 20%, recovery process 14%, and deformity 66%. In this slide, you can see that genotype analysis can be used to express the genotype profile of fry that were having normal operculum and deformity performance. In locus of 2AM2, R21, AS4, and AS15 give gain polymorphism on Barramundi fry operculum deformity. And in table 2, you can see that the average of variation heterozygositas observed is a little more higher, 23.33 is variated. But in operculum deformity, only 8% 0.89. Next slide, please. Next slide, please. Yeah, the conclusion of this research, the study can provide information that the use of feed supplement, probiotic, and vitamin C can be used to recover operculum deformity. The risk of genetic analysis show that barramundi fries with deformity have lower heterozygosity value than normal. I think next slide, please. Thank you very much.
Thank you very much, Bu Hardati.
Thank you very much for your kind attention.
Okay. I will invite next presenter, Pak Laude M. Aslan, or representative. Pak Laude Aslan, or who will represent? Is there any? If not here, so I will invite Pak Muhammad Yamin.
Ya,
Okay.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Please, Pak Muhammad Yamin.
Okay, thank you very much, moderator. Thank you very much for organizing committee. I will share my presentation here. Okay, I'm sorry. My presentation is already. Can you see my presentation?
Yes,
here?
yes, we can see the presentation.
Okay, clear. Okay, thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Please introduce myself. I am Muhammad Yamin. In the next few minutes, I will give a brief presentation about our research result. The title is Survival Rate and Growth Performance of Non-Tetra Parasirodon Inesi in Aquaculture Farm. So the effect of increasing stocking density in water recirculation system. This is our team. I'm Muhammad Yamin, and that's Agus Priadi and the others researcher. This come from research Central Research of Fisheries, National Research and Innovation Agency Indonesia. So for introduction, why we choose non-tetra? Because non-tetra is important species of ornamental fish, especially in market worldwide. And for example, the demand of non-tetra in the US is about 22.7 billion in 1992, and then in the worldwide share market, non-tetra is about demand of non-tetra is about 20% together with guppy. So it's very high, I think, and non-tetra is come from the black and clear water. I'm sorry, my glass I think is bad. And non-tetra is origin from black and clear water, and it's South American Amazon basin. So here the Amazon in America, absolutely, and it's a small size ornamental fish species, only 3 until 5 centimeters long in adult, but they have a very nice color in body, and the important things of non-tetra is most of non-tetra is produced in a fish farm, and fish farm in Southeast Asia. And only 5% collected in the wild in the South America. And from the South Asia, the main producer of neon tetra is Indonesia. And Indonesia, the center production of neon tetra only in Bojong Sari district in Depok City. So Bojong Sari is center production of Indonesia. I think supply for almost all the world. Oh God. In Bojong Sari, for your information, but the main production of non-tetra is culture in the 1 meter dust tank, and they use a stagnant water system. And so with the system, they have some problems, especially infected by disease, because stagnant water system, and then sometimes followed by mass mortality, and then they have a limited land for production, because Bojong Sari close to Jakarta and the price of land is very high, and then they have a problem also with the water by pollution of urban pollution. So to face the problem, to solve the problem, we try to use the water recirculation system to production of neon tetra. So why we use the water recirculation system? Because water recirculation system has a many benefit. For example, is fish is healthier and feed circulation is lower, stocking density is higher, declination water is retaining, most applied in tank absolutely, and then suitable for application in the area that has limited in land and water for intensive farming. And absolutely the last is minimum water change and efficient in maintenance. So the aim of this research is to use the water recirculation system to raise the stocking density of neon tetra. So the methods is we conducted the research in the farmer hatchery in Bojong Sari district, and fish we use is fingerling of neon tetra less than 2 centimeters, and for total is 12,000 individual of neon tetra. And the experiment design was complete random design with the 3-group treatment, and for replication, that treatment was 500 and 1,000 and 1,500 individual per tanks. Glass tank we used to is 100 centimeter times 50 wide and 35 centimeter high, and filled with the 125 liter of water, and all of the tanks is aerated. The recirculation system is containing 4 rearing tanks connected to a filter, and push the water with the submersible pump. And of all filter tank is containing is about 30% of the total volume of water in the 4 rearing tanks, and contained with the 1 meter dacron, 1,000 bio balls, and 1 UV lamp. Parameter ya survival rate, standard length, and bacteria, water quality, body weight, etc. And the analysis we use software Minitab to analyze ANOVA, honest post-hoc test, and then it was analysis also with the descriptive. Here you can see the design of the RAS system for rearing tanks, and water to go to the filter tanks, and then from filter tank go to the UV tanks, and then from UV tank water go to the rearing tanks, and that's the flow of the waters. Here this is In the right
Oh,
side,
sekarang
you can see
mau
the
ke Mr. Troy. Oh iya,
Iya.
iya nanti kontak. Sukses presentasi.
Okay. And the result is for the survival rate at the end of the research, we found all of the group treatment is about 97% survival rate is quite high for all the group treatment. And for the average standard length for all group treatment, I think there's no significant difference between the group. And the total body weight is also there's no significant difference between all group treatment, about 500, 1,000 and 100, 500, 1,500 individual per tank. And the specific growth rate is also there is no significant difference between the all group treatment. However, in the proportion size in half tetra, we found that in 500 individual per tank is dominated by large size. It's about 33.6%, and only few plus 11% small size, small to medium size. But in contrast, in the 1,500 individual per tank, we found dominated by medium to large size, and medium size is about 24%, and only 6.8% in the large size. But for information, all of these sizes is accepted by market, and so farmer don't worry about this product. Water quality in the rearing tanks, we found there is different in conductivity and total dissolved solids, especially in the density 1,500 is quite high compared to 500 and 1,000 individual per tanks. Okay, and in the abundance and type of bacteria, we found that the abundance of bacteria is quite low. On average is low, only in 10 power 4, and yeah, I think that's. Oh yeah, and the important is in the bacteria, we found Vibrio and Aeromonas in the tanks, but there is no there's no bad effect to the rearing tanks. Yeah, for information, beside this research, we have another side. The rest of the fish we put in the aquarium with the same as a normal used to by local people. So with the stagnant water system, but the effect is in the middle of the period research, almost all the fish was die because of attacked by bacteria. So I think that's all. For conclusion, the water recirculation system RAS is suitable to apply in the high stocking density of rearing non-tetra production, and using water recirculation system, the stocking density can be raised to 1,000 until 1,500 individual per tank. It means 2 or 3 times than normal condition in stagnant water system used to by local people in Bojong Sari, and we can got a survival rate until 97% plus and minus. And the application of water, and the last but not least, the application of water recirculation system in the rearing of non-tetra increasing the efficiency of water use because it can reduce some to know water change during the rain period. For your information, farmer with stagnant system, they have to siphon the aquarium 2 or 3 times every 2 or 3 times, 3 days. But in our research, we do not siphon until the last of the period. So no water exchange during the period of research. I think that's all. At the end of presentation, I would like to thanks to Pak Agus, a member of Group of Dakan in Bojong Sari, who helped us during the research period, and the staff of Research Institute for Ornamental Fish Species. I think that's all. Thank you very much for your attention. And now I back to the moderator. Thank you.
Thank you very much, Pak Yamin. And yeah, neon tetra is quite popular, yeah, for the ornamental. Then the producer is come from Indonesia, Pak Yamin, yeah, mostly.
Yeah, mostly from Indonesia.
Okay, now I call again for Pak Aslan or Laude M. Aslan.
Thank you, sir. Now I'm
Yeah,
ready to share my
I-
PowerPoint. Thank
Okay.
you, moderator.
Please stop your presentation, Pak Yamin.
Thank you. Now I do all.
Stop.
How to
Your share
stop screen share? Okay.
Okay. Yes.
Okay, is it
Pak
okay?
Nonasion
Now my
can
PowerPoint
start.
can be seen by the audience.
Yes, Pak Yamin.
Okay.
Pak Aslan.
Okay, let me start to talk about the analysis of sustainable seaweed farming development in our province in Southeast Sulawesi. We are from the Faculty of Fisheries and Marine Science, Palu University. My name is Aslan, and we work along with other 3 counterparts. Yeah, we already know that Indonesia is a significant producer. Now we call also as a global benchmark of seaweed production in the world, particularly the red seaweeds, Cavaficus alvarizii and Acrema. To optimize sustainable production, various multidimensional aspects such as water quality or environment, technology, socioeconomic, government institution, and also technology should be maintained. We think that we cannot improve the production without make an integrated approach or a multidimensional approach to make the seaweed production could be maintained in the near future. And third, there are no sufficient studies available regarding the leveraging factors that contribute to the sustainability of seaweed cultivation in our province. The aims to describe the leveraging factor that contribute to the sustainable of seaweed cultivation in the coastal area of Southeast Sulawesi using multidimensional scaling approach and the red fish application. The study was conducted using both primary and secondary data. The primary data were collected through the World Cafe focus group discussion technique. The FGD with the World Cafe format involved 36 participants, including stakeholders from many actors, especially from the provincial government, research agency, academia, NGOs, the private sector, and other stakeholders. We analyze the data based on the red fish coordination technique modified in red seaweed. This is the multidimensional scaling or MDS approach. This is the multidimensional which is carried out through several stages. First, determining attributes from 5 dimensions. We use ecological, economic, social, cultural, technological, and institutional dimension. Second, assessment of each attribute on ordinal scale. Third, rep-civit ordination analysis using the MDS method through the SPSS program with the aim of determining ordination and stress value. For assessing the sustainability index and status of in each dimension. Fifth, leverage analysis with the aim of determining sensitive variables that influence sustainability, and all data are then analyzed via MDS to determine points that reflect the sustainable position of seaweed cultivation development relative to reference point, namely good or bad points. And for our research, first from ecological dimension, we found that in the ecological dimension, there are many parameters we use, but we should pay attention to aspect: seedling quality and seedling availability. This is a main problem we are still facing with our seaweed farming here, because the seedling quality and availability gradually decreasing. Why? Because all or most or not nearly 100% still use the repetitive seedling by vegetative method. They use more than 50 times using the same method and same seedling without any support from the government to use new seedling. For instance, from the tissue culture seedling or mass selection seedling or grafting method. This is a problem we are facing based on the ecological dimension. For economical dimension, we found that the profit of seaweed farming are still problem. Why? Because price fluctuation, and now the price is very low, and then make the farmer doesn't interest to continue their farming. This is a problem. The second, contribution original local government revenue. This is also a problem because in the last 2 years, the government or local government doesn't support the financial support, or the government should try to buy the dry seaweed to help or to keep the price constantly same for long period. This is very hard to be done by the government. And what about from the social cultural aspect? From social cultural aspect, we found many problems still. First, from educational level. Second, number of farming households tend to fluctuate. Collaboration system also, because we found from our latest recent studies in the coastal area, we found that the farmer always or mostly they work individually. They don't care about the group established by the government. They think that if they work lonely or individually, they can get more profit. This is very interesting. Why the farmer doesn't interest to join the group? And the second, family participation. This is also still weak because they use their own family for their own profit. They don't care about how to make the farmers collaboration between family or among family or between group to other group. And the last is empowerment of seaweed farmers. Empowerment of seaweed farmers also still low because no continue training or extension program done by the government or by dinas. For the technological approach, we found that because most of the seaweed farming located in the remote area, that's make it's very difficult to government or local government to make infrastructure, good infrastructure due to the financial support, low financial support. The second, standardization. That's why in the last 2 years, we already make the standard operational procedure to keep the quality of dried seaweed constantly high. And the third, adoption of seaweed improved farming technique. This is also a problem because, as I mentioned in the previous first presentation, that most of the farmers belong to illiterate or elementary school, or the upper level of their education level is belong to junior high school. It's very problematic situation when we talk about how the technology could be implemented due to the low level education of our farmer. And then last is the availability of seaweed in industry. We have 2 to 3 factories, seaweed factories, funded by the government, but some financial problem, low financial problem, that so the 3 factories doesn't work or cannot be used until this day. This is very sad to say that, very unfortunate situation. And then for institutional, we found that the availability of local rules, the availability of seaweed farmer group, the availability of seaweed extension officer is very low. That's why we cannot get many high improvement due to the institutional problem. And based on analysis of the sustainable index and status of seaweed farming in our province, we found that at least 3 dimensions are still need to be developed. First, socio-cultural, only 42.21, and second is the institutional, 49.15, and the last and very low sustainable index is based on the technological dimensions, only 36.24. And others very ecological is still good because we are very far from the big city. That's why the pollution is not still not become the major problem in our province. And in conclusion, we only mentioned that we mentioned in the fourth sentence that the most important is the institutional dimension, empowerment. We should make more empowerment of extension workers, formation of farmer groups, and the function of local institution and local wisdom. In the last effort, some effort are needed to develop seaweed farming through comprehensive. We focus on the comprehensive collaborative activities based on a 5-dimensional approach. I think that's all. Thank you.
Okay, thank you, Pak sorry, Pak Aslan.
Yeah.
Thank you, Pak Aslan, for the presentation. I suddenly replace Pak Begin as a moderator because he has some errands and quite urgent, so he left this session earlier. So maybe I need to introduce myself. My name is Vita Rumanti Kurniawati, and you can call me Vita. Actually, I will be chairing for the next session, but since Pak Begin has some errands, so I replace him earlier. Now we are in a discussion session. Okay, Ibu Dewi, I think
Thank you, Dr.
I think we have a discussion session now for Ibu Haryanti and then Pak Muhammad Yamin and also Pak Aslan. Any of you have a question from those 3 presenters? Just raise your hand. We have at least we have 7 minutes left for the next session. Any of you have a question? No, not yet. For I think I have for for Ibu Haryanti. Ibu Haryanti, can I ask something? But I
Yes,
asked
okay.
from but I asked from the perspective of non-aquaculture expertise. I don't know anything about aquaculture, but I'm interested about your research because this research, I think you will give kind of solution for the viral disease. So maybe I missed the presentation before, but can you maybe give me short explanation about how significant is this research in what in um, solving the viral disease?
Yes,
That's my
thank you,
question, Ibu
Mbak
Haryanti.
Vita. Yeah, thank you, Mbak Vita. So, in my research is focusing on deformity, not a virus disease,
Mhm,
but
mhm.
the problem for seed production of barramundi. One, uh, there are some problem. One is disease. This is important problem in barramundi culture, especially for viral disease, VNN and iridovirus, and also bacterial disease. This one is also important disease because the infection of bacterial disease will be occur of we call the pit belly disease. The stomach of fish will be swelling like pregnant. This one is because Vibrio harvey. And the
Okay.
third one is deformity. Deformity, a lot of occur in culture of larvae. Yes, because uh, in this case, the prospect is not yet a selective breeding. So maybe there are some gene will lost or something. So very famous is operculum, not properly operculum. So they just have half of operculum in the gill, in gill, so only half. So the gill will be shown with us red color. So this will look like very pity because only have half operculum, not properly operculum, so close properly. No, this one is very a lot of case for these cases. So that's why I I just try for research how to protect the
Fish.
deformity. One
Mhm.
is use a probiotic. We use a Saccharomyces. Saccharomyces is this one is have antioxidant, very high antioxidant, and they will stimulate metabolism in fish, and uh, I researched for 40 days with with the time the operculum they can growing slowly because only 20% they can properly close properly. Only what 15% is in process. More than half of curriculum will be closed, not yet closed properly. This one, but 60%, 66% is still deformity, something like that. So
Oh, okay,
yeah,
okay.
for this is infection is different cases with this with my research.
Okay. Thank you, Ibu
Okay,
Haryanti. I
no
think
problem. You're
my
welcome.
question is about misleading. So this is like between the viral disease and the incidence of deformities. Actually, it's two different things, right? Right?
Yeah, that's
Yeah.
right.
Okay. Yeah. Thank you for the explanation. And
You're welcome.
I have one more question for Pak Aslan. Can I ask Pak Aslan? Mm, Halo, Pak Aslan.
Yes, yes.
Yeah, you mentioned in your previous presentation that there is a dimension. Also sorry if you already mentioned this before. And uh, what do you think if you recreate the this research to other area? I mean, like for example, it's not in Southeast Sulawesi. Do you think this will give a uh, different result? How this will different if you recreate this research to other area which maybe will have uh, different community, for example, because I don't know how's the community in sea farming or seaweed farming. What do you think, Pak Aslan?
You mean that from ecological dimension or
Yes,
what?
from
Yeah,
the ecological and also the social dimension as well.
yeah. For instance, if we talk about the ecological problem,
Mhm,
yeah, first we should understand that Southeast Sulawesi is the mining province of
mhm,
nickel mining. What's the
mhm,
problem for this situation? That gradually our water quality deteriorated due to the heavy metals comes through to the coastal areas. It's a big threats for the future. And the second is that inconsistent action of our government, because we already like have a marine protected area or marine zone or farming zone, but without think for the future need, the government suddenly change the zone for farming activity to be used for the mining or many uh, 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 many activities. Yeah, we thought think about the important to keep the water qualities in the good situation, and then for the social, we found that the most problem from the beginning of the farming, seaweed farming, is we have found that uh, farmers tend to work to farm individually. They are not care about
mm,
to collaborate. They collaborate with other groups if they think if they listen that government will donate or deliver some money. They suddenly make a group, but that group like yeah, we said that it's only for getting some money. After they get money, they don't care about the group. They're again to do the farming activities alone or individual. This is very bad behavior actually, to say that the farmer doesn't believe the other people. They just believe their own family. And the second, we as mentioned before, that low level education, we still found until these days that most of the farmers work as a illiterate farmers. They cannot
yeah,
get new information because they cannot read, they cannot write something, they cannot explore their eagerness. They think about the farming as usual, similar to 50 or 40 years ago. They don't need to change their mind. This is very problematic for our extension or researchers who working in the civic in our province. This is my
okay.
answer. Thank you.
Yeah. Uh-huh. So, do you think that the characteristic of seaweed farming, the socio economic of seaweed farming in your place, is it typical in Indonesia or they're quite
Yeah,
different
actually it's
from
different. Yeah,
other?
because yeah.
Okay.
Thank you. I think it's different. Yeah, because as I mentioned also in our presentation that some local wisdom in different area is very good to support the high production of seaweed. But in our region, I sorry to say that one ethnic
Mhm.
group who work on the seaweed, yeah, have a bad habit. For instance, they
Okay.
do the and they spend a lot of money for unnecessary things. but they easily to get money by doing debt, by asking someone to borrow them money. Yes, that's why we have also a paper about the debt trap of the people working on seaweed farming. This is very bad habit
So I
actually.
think
They spend a lot of
it's
money
about the
for
same in the
unnecessary,
yeah.
but they can get easy
It's
money
about the
by
same in the fishing industry as
some
well.
money by borrowing from the other people. This is very bad for bad habit also for our seaweed farmer. Thank you.
Okay, yeah, that's interesting. Thank you, Pa Aslan, for
You're
your
welcome.
presentation. So that's our second session. So now we're gonna have to continue to the last session. As I as far as I know, we have 4 presenters left. First is Febianti Dwi Kurniawati. Febianti, you here?
Yes.
Okay, and then the second presenter we have Asma Afifah. Asma
Here.
Afifah, okay. And then the third one we have Fadel Khairul Akbar.
Yes.
Okay, and then the last one we have Ismi Istianah. Istianah.
I'm here.
Okay, yeah, we have, so it's a complete presenter. So to make it the time more efficient, so I'd like Febianti to start
Yes.
your presentation, please. So the screen is yours.
Thank you for the time. I will present the result of my research on the topic bioactive compound, antioxidant activity, and phycocyanin content of culture and commercial spirulina pregnancies. The next slide. Please next slide.
Next, please. Next, please.
Sudah sekarang di apa namanya? Rekening apa namanya? Yang 12.000 itu
can you please mute your microphone, please?
Oke.
Yeah, that's okay. Thank you.
The outline that I will present today, introduction, material, and method, result and discussion, and the last conclusion. Next, please. Next.
I think there is a problem with the
Yeah,
operator.
okay, I will share screen my PPT by
Oh yeah, that's good. Yeah, thank you.
Yes, Spirulina platensis is a blue-green algae that type of cyanobacteria. The Spirulina platensis have a cellular and filamentous. Spirulina have high nutritional content such as protein, lipid, and carbohydrate. Spirulina can grow optimally in the pH range of 9 to 11. The utilization of Spirulina platensis promising sort of food and feed. The pigment content in spirulina is chlorophyll, phycocyanin, carotenoid. Spirulina have two unsaturated fatty acids such as alpha-linolenic acid and gamma-linolenic acid. Spirulina platensis is capable to producing the antioxidant, anti-diabetic, anti-plasmodial, and anti-cancer bioactive active compound and bioactive in spirulina. The influenced by temperature, light intensity, pH, salinity, and nutrient. Commonly, media that used to cultivation of spirulina, walnut, and zagreb. This research aimed to determine the active compound, phycocyanin content, and antioxidant activity of the Spirulina platensis cultivated using fertilized media, and also to compare cultivated spirulina and commercial spirulina. These some material we use to analyze in this research, and this some equipment we use. Method of culturing spirulina. First, we will culturing of spirulina in aquarium 50 liter in indoor with fertilized media. We use light intensity and photoperiod to 24 hours in salinity 20 ppt with aeration non-stop 24 hours. First, we will refresh of spirulina patensis and then culturing. We can resulting the spirulina with wet biomass and then we drying oven in oven with 40 degrees Celsius in 24 hours, and we have dry biomass. So we continue the extraction of the spirulina in maceration to have spirulina patensis crude extract, and then we can analyze active compound, total phenol content, and antioxidant activity. And some of dry biomass we extract with freeze thawing method, use phosphate buffer to analyze phycocyanin pigment. And this the result. The highest of OD value on day 14 is 0.3. The highest of OD, this indicates that the spirulina can grow optimally. Susila et al. 20 and 20 state that his research have OD value is 0.5 in day 14. Is the yield of spirulina patensis. We have 0.14 and the yield spirulina patensis on glass tube have 0.22 and on aquarium 0.14. Search of Wulandari et al. 2016. Growth and biomass production of spirulina patensis are influenced by temperature, light intensity, photoperiod, salinity, and growth media. Have crude extract of spirulina patensis in the color of extract cultivated spirulina is brown, but the color of extract in commercial spirulina is blackish green. That means the cultivated spirulina have more good sample to be analyzed. The yield of spirulina can impact to water content. We analyze the maximum water content requirement is 10%. Is the active compound we have in spirulina extract. The cultivated spirulina detected saponin, steroid, flavonoid, and phenylhydroquinone. That means same of the Aenia 2017 research, but in spirulina commercial just have saponin and steroid. Total phenol content. The total phenol content can influence the antioxidant activity. More higher of phenolic content can influence the antioxidant activity more higher. The factor influence to total phenol content is for culture media and drying method. In cultivated spirulina extract, we have 15, and commercial spirulina we just have 9. This antioxidant activity in cultivated spirulina extract, the IC50 we have more flower. That means the cultivated spirulina extract have the antioxidant the activated the cultivated spirulina extract have antioxidant activity more higher than commercial spirulina. This phycocyanin is the blue pigment in spirulina, the result of concentrasi phycocyanin using crystallizing method with aqueous as a solvent is 0.2 of the research Embrio et al. This phycocyanin extract factor that affect the content of phycocyanin is media, light, drying method, environmental condition of spirulina. The uncultivated spirulina have a yield phycocyanin more higher than commercial spirulina potencies. On conclusion, cultivated spirulina potensis extract active compound, total phenol content, and antioxidant activity higher than commercial spirulina. Cultivated spirulina potensis also contain a higher concentration and yield of phycocyanin compared to commercial spirulina potensis. Thank you.
Okay, thank you. So this is the presenter is Mbak Husnul then.
Yeah.
Yes, okay. Thank you for your presentation, Mbak Husnul. And now we move to the second presenter for this session, which is Mbak Asma Afifah. Okay, the
Yes,
screen is yours.
I will share the PowerPoint first. It's already shown in the screen, right? On the screen.
Yes, you just need to slideshow.
Okay, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Sorry, it's not slideshow yet.
Oh yeah.
The mode is not in a slideshow. Okay,
Oh.
yeah, that's okay.
Okay. So, good afternoon, everyone. I'd like to introduce myself. My name is Asma Afifah. I'm from Aquatic Product Technology, Faculty of Fisheries and Marine Sciences. I'm here today to explain a bit part of the result of my research. My talk will last about 10 minutes, and without any further ado, I will start my presentation. Under title, Physical Characteristic and Phycocyanin Content of Spirulina potensis cultivated using artificial seawater. So this talk is divided into 4 main parts. I will start with introduction. Secondly, I will be talking about the materials and method. The third point is the result of my research, and the last is the conclusion. So let's start with the introduction. Sorry. Oh, Spirulina potensis is a single-cell spiral 2 green algae contain nutrient component and active compound. As we can see in the screen that spirulina contains 70% protein and then 15 until 25% carbohydrate and 8% others. And spirulina potensis has active component, bioactive compound such as phenol and then triterpenoid, steroid, flavonid, and saponin. And it can be used as anti-cancer. And then spirulina has biopigment too, like chlorophyll, allophycocyanin, picoerythrin, and phycocyanin, which is phycocyanin is the most important protein of spirulina potensis that has antioxidant and radical scavenging activity. Spirulina potensis is generally used in the food industry and then cosmetic industry, medicine, fuel, and aquaculture feed. So, and then the environment for cultivation needs to be considered, such as the habitat of the microalgae. Spirulina cultivation in general can use the fresh water and then seawater and then brackish water. Selection of the environment is essential as different as water provides different nutrients and environmental aspect that could affect the growth of the cell of spirulina. Several factors that can affect cell productivity are temperature and then pH, salinity, light intensity, macro and micronutrients. So spirulina cultivation can be done using artificial seawater as an environment to make the cultivation easier if the cultivation location is far from seawater sources. From the previous research, Spirulina platensis grew better in artificial seawater than Chlorella vulgaris. So the use of salt in spirulina cultivation is suspected effect on pigment and nutrient component. So the objective of this research is determine the effect of the various artificial seawater on the Spirulina platensis culture. And then the research benefit to provide information on the cultivation of spirulina using artificial seawater. The third part is material and method. This is the materials that used in this research, as you can see on the screen, like spirulina inoculant and then Epsom salt, table salt, Krosok salt, and then artificial seawater salt, Quades, and walnut. And then this is the equipment that used in this research: jar, and then hose, aerator, refractometer, and then nylon mask, centrifuge, spectrophotometer, and chiller. So the research procedure is the first cultivation, harvesting, and drying of Spirulina platensis biomass, and then the second is extraction of Spirulina platensis and phycocyanin. So this is the cultivation of spirulina. Cultivation begin with sterilization of all equipment using UV light for 45 minutes. Spirulina cultivation was carried out in 10 liters capacity jars using walnut media and with addition of different salt. With you can see on the screen that water and then Krosok salt and then water plus table salt, and then water plus artificial seawater salt, then water and Epsom salt, and for control we use fresh water and seawater. Is all the treatment is 15 ppt. So the cultivation, the aeration and light was carried out continuously, like 24 hours for 14 days. Sampling was carried out by measuring the optical density of the cultivated spirulina. Harvesting of Spirulina platensis biomass was carried out after the optical density more than 0.5, reach more 0.5. So after that we have the growth rate, and then for the harvesting of Spirulina platensis was carried out by filtering using nylon mask, 200 microns, and then we get the biomass. And for drying, we use a chiller in 10 Celsius degrees for 48 hours, and then we get the dried biomass, and then we get the analysis water content, yield, and function. So the result is Here, the optical density. Optical density value can be assumed to be the number of cells of microalgae growing inside certain period of time. Growth of Spiruna patensis can be determined from the color change in the cultivation, which indicate an increase in population number of cell density. Spirulina culture had 4 crowd phase. Like this is like the lag phase. Lag phase is like the adaptation phase, and then stationary exponential phase. This is increase a lot, and then the stationary phase, it's just like here stuck, and then the dead phase, which is the cell the optical density decrease, and then the the highest optical density is on table salt in day 10, 0.588, and then for the longest cultivation in Epsom salt, day 14 here on the like 0.5 12. For the exponential phase is the best phase for harvesting and producing the best bioactive component. And then high salinity also can affect on the growth of microalgae, because the cultivation environment is dominated by the presence of Na plus N salmine, which can disrupt the osmotic balance between inside of the cell and its external environment. And then for the yield, we can see the highest yield is the Epsom salt with 3.4 grams in harvested on day 14 cultivation on its exponential pace, but for the other treatment harvested on the dead phase. So nitrogen is an essential component of structure and function of protein and alga cell. On the growth of spirulina, the mangan in salt also plays a role. The physical of microalgae can be seen through the growth and density of their cell. Sodium or Na plus and potassium K plus are two components that are not replaceable in spirulina growth media. And then the micro and the nutrients is important too. So this is for the other treatment. If they were harvested on the exponential phase, it is possibly it is possibly produce the higher yield than when we harvested on the dead phase. And factors that influence the biomass yield of spirulina patensis are environmental conditions such as the availability of nutrients, and then light intensity, pH and carbon source. And then for the picocyanin, we can see here the high compared between all the salt, artificial seawater salt contain more. I think it's a bit more nutrients than the other salt. So
can you speed up, because the time is actually up?
Okay,
Please
so
speed up
this
your
is
presentation.
okay. This is for the appearance of the phycocyanin for each salt, and the concentration here. So if the purity index of the precaution is greater than 0.7, then it is classified as food grade. And then if 3.9 is classified as reactive grade, and if it is greater than 4, it is classified as analytical grade. If we wanna increase the purity index, we can do purification. So the conclusion is the use of artificial seawater in Spirulina platensis cultivation has an effect on the Spirulina platensis culture, and then artificial seawater solution as artificial seawater is the best treatment on this study and can be used as cultivation environment for Spirulina platensis. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Mbak Asma, for your presentation. And next is the third presenter, Fadel Khairul Akbar. The screen is yours.
Share screen my PowerPoint. The screen is displayed. Well, hello.
Yes. Yeah.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. First of all, thank you for your presence and being here. Let me introduce myself. My name is Fadil Akbar from Department of Aquatic Product Technology, Faculty of Fisheries and Marine Science, IPB University. I'm going to bring our research with the title Characterization and Antigenicity of the surimi and kamaboko protein of Japanese redfin bream, Nemipterus japonicus. Food allergy is a specific immune response that causes an abnormal reaction by the body's immune system to several food that are allergen. Food allergies affect approximately 2.5% of the population worldwide, and their prevalence is always increasing. The prevalence of food allergy in Indonesia 5 until 11%. Food allergies can be caused by lactose, egg, nut, shellfish, trans fat, gluten, shrimp, soy, and also fish. In Asia, the highest prevalence of allergy generally come from processed fish food. Okay, the source of allergies in fish is usually found in protein because fish contain high level of protein. Protein that have been identified as allergen include beta-parvalbumin, beta-enolase, and aldolase A. Parvalbumin is known as a major fish allergen because most of fish allergic patients are sensitive to parvalbumin, while beta-enolase and aldolase are considered minor allergen. One of economically important fish that is often caught in Indonesia is Japanese redfin bream, or Indonesia called kurisi or terisi. Kurisi is usually used raw material for making surimi. Surimi is a fish protein concentrate obtained by washing fish to remove blood and most the fat, enzyme, and sarcoplasmic protein. Processing can change the structure and chemical properties of proteins. There will be potential changing the allergenicity of a food ingredient, especially for certain allergens such as parvalbumin. The response to change in parvalbumin content can vary depending on the type and stage of processing carried out. Research by Nugraha et al. Showed that the washing process in making surimi can reduce the parvalbumin content. Abidin research 20 and 23 show that the washing process in making surimi can reduce the parvalbumin content using SDS page as a detection method, but the detection limit is still high with the background as explained. An attempt we made compared to meal-free protein from meat protein during the surimi making process and carry out an ELISA kit to detect parvalbumin protein instead SDS page because the detection limit was still high. Okay, the method, this research was conducted from August to November 20 and 23 at any laboratorium. The method used in the research consists of the process. First, the process of making surimi or extracting myofibril protein, which consists of washing and filtering. And second, making a kamaboko from the surimi that has been made. Okay, for the research procedure, you can see the PowerPoint display. And this analysis I use in my research, proximate analysis, turbidity, surface hydrophobicity, molecular weight, determination chemical bond, and allergenicity. For the result, chemical composition of Japanese thread green fish determined using proximate analysis. The result obtained from the proximate analysis of Kurisi show that the fat content of Kurisi fish is 0.76%, which is included in the low fat fish group, and the protein content in Kurisi is 18.73%. The protein content of Kurisi fish is relatively high. Fish that have a protein content 15 until 20% are classified as a high protein fish. Factors that influence the chemical composition of an organism are age, environmental condition, size, freshness, and length of storage. Okay, the protein concentration of Kurisi fish meat was measured using a Bradford method. The measurement of dissolved protein decreases after the washing process in making surimi. The washing process in making surimi can reduce the protein content, more precisely the sarcoplasmic protein content. Sarcoplasmic protein content is a protein that soluble water, so every process of making surimi will reduce the soluble protein content. Next, surface hydrophobicity. The hydrophobic group of protein molecules characterize the tertiary structure of the protein. The hydrophobic interaction force help in maintaining protein structure, also influence the stability and functional properties of myofibril protein. It can be seen from the result, then the BPB bound value increase during the process of making surimi. Surface hydrophobicity reflects the emulsifying property and is related to the solubility of the protein. The more BPB bound to the myosin molecules, the more hydrophobic side on the protein structure. This increase in BPB bond value indicate the unfolding of myofibril protein and exposure of non-polar amino acid residues to their surface. Nevertheless, the degree this unfolding of myofibril protein depends on factors such as ionic environment, extraction condition, and intrinsic factor of the proteins that the difference in hydrophobicity. Next, turbidity. The higher turbidity might be due to the continuous formation of large protein aggregate that cause light scattering and impede light transmission. The disparity of turbidity probably might be due to the difference in particle size and or rate of property of protein aggregation. The presence of large aggregates formation is apparently equation for the formulation of good elastic gel. It has been reported then the difference in amount hydrophobic and dissolved bond might also be one of possible reason for difference in aggregation and can certainly lead to change in turbidity. Next, chemical bond. The disruption of hydrogen bond between water and protein molecule lead to unfolding of protein molecules, and this result to gradual decrease in apparent viscosity. The presence of more hydrogen bonds is possibly to be a reason to more bond surface hydrophobicity in kamaboko. And the profile protein kinase is the most commonly found enzyme, followed by aldolase, dehydrogenase, isomerase, and enolase. And most of the F-des enzyme have a molecular weight of 37 until 50 kDa. Other protein dissolved in the washing process are phosphobumin. The protein content of surimi influenced by the washing process, which can remove sarcoplasmic protein, fats, and the other nitrogen compound from mesh fish meat. The washing process in the study was identified as being able to reduce most of the main protein, as seen by the similarity of protein profile in surimi and washing water. Allergen content from SDS page result. The result of this study show that mean that has undergone the washing process show a decrease in the intensity of protein band tau the parvalbumin, beta enolase, and aldolase in the range of 10 until 13 kilodalton and 37 until 50 kilodalton compared to the initial prevalent meat before washing. Next, allergen content from ELISA kit. Detection of allergen protein in surimi sample was carried out using the enzyme-linked immunosorbent assay or ELISA method. The ELISA kit used is the Agraquan fish ELISA plate. which use the interaction of specific antigen antibody to detect and quantify allergen with a photometric measurement. The advantage of this method are that it obtains quantitative result, has high sensitivity and specificity, is fast, and allow for testing several samples at once. Allergen measurement we carried out using an ELISA reader with wavelength of 450 and 630 nanometer. The result obtained from ELISA kits show the allergen content from fresh meat to kamaboko has decreased. The intensity of parvalbumin banned in puree fish meat decreased after washing process. This parvalbumin is carried by the washing water, which can be seen from the high parvalbumin intensity value in AP1 and AP2. Research by Kurata et al. 2017 using the ELISA method found that the concentration of parvalbumin in Alaskan pollock meat decreased by 87% after 2 washing and decreased to 99% after 5 washing. Conclusion: and the allergenic protein content found during the processing surimi include parvalbumin, aldolase A, and beta-enolase. Using the ELISA kit, get more precise result to see a reduction in allergen content. The allergenic protein of Japanese redfin bream meat decreased after the washing process. The decrease in allergenic protein content occurs as the protein dissolves sarcoplasmic. Protein characteristic change during surimi processing, starting from chemical bond, turbidity, and surface hydrophobicity. In suggestion, in the next research can use the modification to the surimi processing process as an effort to maximize the reduction in allergic protein content. Then it's necessary to use sequencing with LCMS to ensure that the protein found is targeted allergenic protein. Thank you.
Okay, thank you for the presentation. So now we are coming to the last presenter, which is Saudari. Ismi Istianah. Ismi Istianah. Mbak Ismi? Ya. Ibu Ismi atau Mbak
Thank
Ismi?
you.
Yeah, you may start the presentation. Please, the screen is yours.
Okay, I will share my presentation. Can you see my presentation?
Yes, it's clear.
Okay, thank you. Good afternoon. Thank you for Dr. Vita as the moderator at this session, and all of the committee of the Eis Embrio International Symposium 2023. I'm Ismi Istiana, and I will do my presentation about protein analysis and cellulase enzyme activity of marine endophytic fungi, Aspergillus terreus. These are my supervisors, Win Chen Li, Aria Tarman, Sugeng Heri Suseno, Roni Nugraha, and also Ilza Effendi. Fungi that symbiotically coexist with marine organisms, such as seaweed, swans, and mangrove, are known as marine endophytic fungi. This is the diversity of marine fungi habitat, and there are approximately 1.3 million species of endophytic fungi in tropical ecosystem, representing a recent surge of bioactive compounds. And these marine endophytic fungi are microscopic fungi that live intracellularly within plant tissues without damaging their host plants. And we can isolate this from green seaweed, or seagrass, or mangroves. And mangrove that contains endophytic fungi, namely mangrove endophytic fungi. This one is one species of mangrove called Soneratia alba, and we can find it in Southeast Sulawesi, which is one of the provinces in Indonesia with a potential mangrove ecosystem. And the mangrove forest
on Buton Island in Southeast Sulawesi belong to the category of coastal forests that can thrive in high salinity condition and also extreme environments. It possesses antibacterial properties that can be used as a natural source for the generation of antimicrobial therapeutics. And this is the biological activities of fungal endophytes. And if we use these bioactive compounds, it has high level of protein as well as various type of the level of amino acid, fatty acid, immunostimulants, antioxidant, and also pigments. It also the renewable sources and sustainability, and it does not produce the toxic byproducts. And marine endophytic fungi and its bioactive compound can be application in biological control agents for Coledotrichum acutatum on chili plants, and also antibacterial against livestock pathogens, and application in aquaculture sector for increase the fish and crustacean health and also the human health. And the proposed life cycle of using this fungal EPS is fungal fermentation, and then EPS extraction, also purification. After that, the characterization, or the modification process, and the end is biomedical application. So this research is conducted on October 2022 until 2023, and then the research location are in Laboratory of Microbiology, Department of Aquatic Product Technology, and then the Advanced Research Laboratory, IPB University, and also iLab in National Research and Innovation Agency, BRIN, and also Department of Chemical Engineering, National Chungcheong University, Taiwan. My research hypothesis is marine endophytic fungi have extracellular enzyme and bioactive compounds as marine natural products. So these are the research procedure. The first is preparation for SDSPH, and then metabolite analysis using HPLC, and also cellulase enzyme activity. The first is preparation for SDSPH. We must do the pre-culture of isolate from Aspergillus stereus species and doing the filtration using Wetman filtered paper to separate the mycelia and the culture, and also making up and down solution for the SDSPH. Afterwards, running to the gel electrophoresis and at 60 and 120 volt, and also gel analysis. After that, we do the metabolite analysis. We are preparing the extract of Aspergillus stereus and then inject 1 millisample to the HPLC tube and run the sample in the HPLC machine for the 25 minutes using 87 column. And then the cellulase enzyme activity, doing the pre-culture of marine fungi isolate using CMC media, and then taking the culture and centrifuge at 500 for 15 minutes and at room temperature, and use the supernatant of measure the enzyme activity and also the characterization of cellulase enzyme. The result and discussion. This is the SDSPH result. The left side is the marker, and the third sample is consisted of the liquid of the culture of Aspergillus stereus. The SDSPH result is 17 kilodalton molecular weight, and it has the similarity with endoglucanase enzyme. The zymogram studies reveal the presence of the 7 isoform for the endoglucanase that having molecular weight corresponding to approximately 18 until 50 kilodalton, and endoglucanase with molar mass of 8 until 5 until 7 kilodaltons show higher affinity for low polymeric cellular substrates and oligosaccharides. And then this is the HPLC result for crude extract of Aspergillus stereus. Now the highest is the concentration of glucose, And then another peak show the organic compounds such as succinic acid, that we know that succinic acid can serve as an antimicrobial agent due to it can suppress the growth of the pathogenic such as Staphylococcus aureus and also Escherichia coli. And then this is the standard curve of glucose. And we know that for measurement the cellulose enzyme activity was determined by converting absorbance values obtained from the standard glucose concentration, then calculated by using a formula. This is the standard curve of glucose with Y example absorbance and X is concentration of glucose. With the regression, this is 0.9911, and then the enzyme activity. Sample A is for static condition of the culture, sample B is for shaker condition, and the measurement from day 1 until day 6 show that the enzyme activity of A optimum at the day 4 and the enzyme activity of B optimum at day 6. For the pH and the temperature of Aspergillus cereus, the optimum pH show that the enzyme activity of A optimum at pH 4, and then the optimum temperature of A optimum at 60 degrees Celsius. So the conclusion of this research is analyzing protein from marine endophytic fungi Aspergillus cereus using SDSPH has 17 kilodalton molecular weight band of protein, and it indicated to extracellular cellulose enzyme. And the bioactive compound from marine endophytic fungi Aspergillus cereus can be measured use HPLC. It has compound that have some retention time with glucose, succinic acid, and also acetate. And the measurement of the cellulose enzyme activity gives the optimum enzyme activity result on the day 4 and 6. pH optimum of the culture of Aspergillus cereus is 4, and its optimum at 60 degrees Celsius temperature. Thank you for all the committee of the 6th Embryo International Symposium 2023. I also want to say thank you to the Ministry of the Education and Department Aquatic Product Technology, Faculty of Fisheries and Marine Sciences, and also CCRMS, IPB, and also National Chung Cheng University. Thank you.
Okay, thank you for your presentation, Mbak Ismi. So now we are entering the discussion session. Any of you want to ask for those 4 presentations? Please raise your hand. Any question from the participants here? Any question? No, not yet. Is it because very clear or else? Okay, I think I have several questions for participant for the presenter here. But if any participant later have another question, just please raise your hand or give me a shout through the chat, or maybe just open up your microphone. Maybe my question will be the very basic one as well, because I'm not from the this field as well. So first, I have a question for Mbak Kusnu. You make a comparison between spirulina from the culture and also the commercial one. Actually, for me, myself, I'm not really understand what is the difference between commercial spirulina and also cultured spirulina, and why you need to develop the cultured spirulina. That will be my question. And then for Mbak Asma, you develop artificial seawater, which is interesting. And you mentioned before that this will be a solution or to solve the problem if you want to cultivate. Is it cultivate? Yeah, cultivate the spirulina, but it's quite far from the sea, so you can use the artificial seawater. But the result is surprisingly using the artificial seawater is way much better, right? Than the using the normal normal seawater. So my question is, do you think that this is still used? I mean, it's still okay to use. I mean, better to use the artificial seawater or just use the normal seawater. I mean, maybe like, do you think that seawater is not useful anymore to cultivate the spirulina? I don't know. Maybe I need your perspective, so that's why I asked this question. And then for Fadel, uh, Mas Fadel, yeah.
Yeah.
Is there any best way to wash the fish then? I mean, like to produce the less allergenic uh, fish or surimi or whatever it is, because this is said that it is coming from different procedure of washing, and then you can produce a better fish because without the allergenic component. Yeah. And then last but not least, I have one question as well for Mbak Ismi. Uh, this is kind of you you analyze the protein and also enzyme from the fungi. What does it mean that? Is it mean that this is a good, moderate, or best fungi? I'm for to support its function in your area. That's will be my question. So yeah, can you please answer? Maybe starting from Mbak Kusnu.
Okay, thank you for the question. Why I compare between cultivated spirulina and commercial spirulina? This research to look where the most better between cultivated spirulina or commercial spirulina, because in commercial spirulina we don't know what the method they do in cultivated, in drying, and for the result, we know that the cultivated spirulina in laboratory have component active and total phenol and IC50 more better than spirulina commercial.
Okay, yeah. So that's it.
Yeah, thank you.
Okay, thank you. And now Mbak Asma. Yeah.
So it's actually still necessary. I mean, the seawater is still necessary, but for the one who maybe for the cultivator who wanna cultivate and but the location is far, this is can be one solution for that case because usually like to transport or logistic to bring the seawater to the location that we will cultivate it like pricey, and then well actually we can cultivate with fresh water, but the fresh water, the result from the fresh water not as good as used seawater or artificial seawater. And then so fresh water, we can make artificial seawater from fresh water and then salt, like we make a solution, then we cultivate on that environment, and surprisingly the result is good, and then with this method we can minimize the what but wait, we can minimize the contamination,
Okay,
like
yeah.
yeah, because and then we can adjust like the nutrient, and then something that, I mean, the result that we want, we can adjust the nutrient and the salt, because more when we take the seawater deeper and deeper, the mineral will like we got a lot like we got higher salt content, and then mineral content, which is like ash content, so ash content. So it's I think it's not really good for our health, like the salt, if the salt content is high.
Okay. So
Yeah.
when you make the, do you mean like for the normal seawater, it should be taken from the like very deep water, or you
Yeah,
can
well,
just
actually,
use the surface water?
no. So because the surface water is a lot of contamination
So it's deeper
from the
in the, yeah.
yeah, from the other environment, like from so the water, so tip like maybe like the yeah, the
One and a
seawater,
half meters, 1.5 meter deep
yeah, like
depth,
2 meters.
or 2
Yeah,
meters. Okay.
yeah. I
Okay. Yeah.
yeah, I took that before, like 1.5 meters or 2 meters.
Okay. Yeah. Thank you, Basma.
Pleasure.
Mas Fadel.
Okay, to get fewer allergen, you can increase the number of voices you can add some salt, such as an NaCl or CaCl2, because the addition of salt can bind to myofibril proteins, so that when washing is carried out, the only protein that are washed will be the sarcoplastic protein. Uh, I was intern in surimi processing plant. In this procedure, this plant use salt, NaCl, and CaCl2 for better gelling agent gitu. Struktur gel and the sarcoplasmic protein much better, and so the allergen in the result favor than to get allergen in the result favor than the before.
Okay. Did you just say that if when washing the fish, it's better to add salt? Did you just say that? To add salt?
Yes, when
Mm.
when washing, added some salt is in the previous research can better we then not added salt.
Okay. Is it implementable? I mean, like, is it doable for other kind of fish? I think because maybe for the next if I want to wash the fish, I mean, like, it's better to put salt or etc. Or what do you think? What is your suggestion?
Yes, the other fish can be added the salt or uh, generally NaCl because it's NaCl in the product apa itu sebutnya? Wait. Uh, Added salt in the plant is the in the plant usually added salt like NaCl. So
Mhm.
added salt in washing is good for allergen in the result.
Okay, yeah, that's a good information for me personally. Okay, thank you, Mas Fadel. Last but not least, Mbak Ismi.
Thank you, Ibu Dr. Vita, for the question. I will answer that this research actually is one part of my dissertation research. So before this, I also did the essay about antimicrobial, antioxidant, and metabolite profiling, also the mechanism of action. And I have been done the isolation for 160 samples, the marine endophytic fungi from the seaweed and seagrass and also the mangrove. But the selective isolate and the best activity is on mangrove endophytic fungi. So I do the research further about this. I do the enzyme analysis, and this enzyme analysis show that it has the good potential as a cellulolytic enzyme as a marine natural products for the biomedical application. Thank you.
Okay. Yeah, thank you. So I think that's the end of our session. Thank you for a very, very nice presentation, Mbak Husnul, Mbak Asma, Mas Fadel, and Mbak Ismi. Uh, as the person from a different kind of background, I get a lot of uh, interesting information from this session. And since this is the end of our session, so after this, you may back to the main room because we will have another
Excuse me, Miss.
closing
Uh,
ceremony. Yes?
before we leave the breakout room, there will be a
Mhm.
photo session for the last time
Okay,
in this
yeah.
breakout room. So, everyone,
Okay,
would you
yeah.
please
Can you
mind
please
to
turn
turn on the
on the
camera?
camera, please? Who's gonna
Maybe
take the
we,
picture?
me.
Okay.
Maybe we will wait for like 10 seconds to everyone to prepare your camera. Okay. Okay. I will conduct the photo session. 1, 2, 3. Last one. 1, 2, 3. Okay. Thank you, Miss Vita.
Okay, thank you for the committee as well. So thank you for everyone. We meet you again shortly in the main room. See you and have a nice day.
Me.
Bye. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank
Bye. Thank
you.
you, Ibu Dr. Vita.
Thank you. You're welcome.
Okay, ladies and gentlemen, how was the breakout room? How was the oral session? Was it informative? Was it fun? I hope it is. For now, we have now reached the last agenda of the event, which is the Best Presenter Award announcement. The winner of this symposium, Best Presenter, will be categorized into student and non-student presenters, and also there is a category for Best Poster Award. The aspects that affects the score, which is determined by the assigned moderator for each session, are punctuality, presentation skill, informativeness, clarity, readableness, flow, and question handlement. So, without any further ado, let's announce the winner now.
Okay, the winner for the Best Presenter Award for non-student category is, congratulations to Mimi Irani from Faculty of Fishery and Food Science, University of Malaysia, Terengganu. For non-student category is, congratulations to Mutiara Putri Rahmadatul Aji from Faculty of Fisheries and Marine Sciences, RVD University. Congratulations. And lastly, the winner for the Best Poster Award is Andini Nabila Putri from Faculty of Fisheries and Marine Sciences, RVD University. Congratulations. And for the winner for the Best Presenter Award and also the Best Poster Award today, please kindly send your address to the committee on the direct message.
National Symposium.
Thank you for your active participation throughout the whole 2 days.
I am